UK, I think we are living in the second lockdown. The lockdown isolates people, depresses people, and brings economic downtrend. Let's look at some of the figures we have from government yesterday. About 300,000 people are tested for COVID every day in UK. Of those, they're picking up between 23,000 to 25,000 positive cases. Death rate yesterday was 378, taking to the grand total of 48,475 people. Very worrying. So what shall we do? So let's listen to our expert today. Now, our expert is Dr. Ravi Gupta. He's son of our good old friend Nagendra Gupta. Ravi Gupta is a professor of clinical microbiology and therapeutic immunology in Cambridge. He was graduated from Cambridge in '97. He had his higher education both in Oxford and Harvard University in USA. Subsequently, he achieved PhD in virology in UCL. Since then, his main interest was HIV treatment, both in UK and in Africa. But recently, his interest turned towards COVID. In COVID, he's concentrating on plasma treatment and remdesivir treatment. Now, Ravi, most of us in this audience, we crossed 60 years and we are eagerly waiting for vaccine, and I hope you can deliver that today. Ladies and gentlemen, let me invite Ravi to address on COVID immunity and vaccine therapy.
Thank you so much for that introduction. That's very kind of you, Uncle. And it's really nice to be able to speak to the group. I think it's really great that you're doing this, and very exciting. So in terms of COVID, I mean, my work in COVID is stretched through a number of different things so far. I mean, right at the beginning, we started trying to do rapid diagnostics because the test was too slow in the hospital here. So we implemented and tested some really quick tests for nose and throat swabs and also antibody tests. So, but we're not going to talk about that today. I think what people are really interested in is vaccines and, of course, how we get immunity to COVID-19. So these are the big questions, I think. So I think we'll focus on the talk on this at the moment, and I'll try and introduce some things that we've been doing in our lab around this, which feeds into immunity and vaccines. Because, of course, to make vaccines, you need to know what the pathogen is. You need to know how it mutates, and how sensitive it's going to be to the treatments that we use or the vaccines we make, because the vaccines are designed against specific strains usually. And a lot of our expertise from HIV has helped us in COVID. I mean, we would be nowhere without HIV research in COVID-19. All these monoclonal antibodies you talk about or hear about, these were all developed initially for HIV, and all of that knowledge has transferred over to COVID-19. So even though AIDS is not something that people are interested in this country and yourselves, without AIDS, this would not have been, we would not have been anywhere near a vaccine at this moment. So that's really important to emphasize. So, okay. Let's start with a bit about this virus, okay, because to understand immunity and vaccines, you need to know about the virus. So this is a little picture on the screen here. Can everyone see the pointer? Maybe yes. So here, this is the virus particle, and it starts on the inside. This is the genetic material of the virus. It's RNA. And it's really quite big. It's about 3 times the size of the HIV genome. So it has it encodes quite a lot of different proteins and has a lot of functions. And this is typical of coronaviruses. That's surrounded by a lipid layer, which the virus takes this lipid layer from our cells when it is produced from a cell. So this lipid around here is our own, is from us. and embedded in that lipid layer are different proteins. One is called E for envelope. There's also an M for matrix, membrane glycoprotein. But the one that people are very interested in is the spike protein S. And the reason that's, this is blown up here on the right. This is a blow-up picture of what that structure looks like. It's composed of 3 units, and that's why we call this a trimer. It's got made up of 3 individual units which come together. These you can see these balls are in each particle, in each monomer to make a trimer. And so the reason people are interested in this is because it is the part of the virus which. Oh, I'm just wondering how I move. Okay, fine. So this is the bit of the virus which enables the virus to enter the cell. So this protein here will bind to ACE2 on the surface of a target cell, like your cells in your nose or your throat or your lungs. And once this binding happens, the virus can enter the cells. We may talk a bit more about that later. But the reason spike is also important is because our antibodies, and this is a sort of simplified antibody drawing, our antibodies are actually targeting this protein on the surface. So when we make an immune response, a lot of our antibodies are directed against the spike protein and different parts of the spike protein. And we think that if we make antibodies, we can be protected against infection, and that's why vaccines are largely targeting. Vaccines are largely trying to make the body produce antibodies against the spike protein, but other proteins as well. So it's a very important part of the virus to understand. And you may have read about the mink mutants. They're about to cull millions of animals because they think that a mutation in this spike protein has arisen in those animals and is transmitting to humans now. So the worry is that these mutations might compromise the ability of our vaccines to tackle the virus. So that's really quite important. Okay, so just very briefly, this is just a diagram to show you really what you probably already know and have read, but it's important because it tells you how the immune response actually starts working, and the fact that the immune response might be responsible for some of the damage we see. So right at the beginning, you've got the time course here on this part, and up here you've got the severity of your illness. And in the first week to 10 days, you get this obviously large amounts of virus in the nose and throat, and sometimes and then the lungs. You may have mild symptoms, or you may have quite severe symptoms, but usually dry cough, headaches, diarrhea, fever, these are all key signs. And this is marked by high levels of virus replication. We can see a lot of virus coming out in patients during this time. And in this period, potentially, this is where remdesivir and drugs may have a role, including monoclonal antibodies or convalescent plasma infusions. This is the stage where the virus is doing a lot of the damage. Then you get this other phase where the immune system is obviously the immune system is triggered very early on, but it takes a while to develop things like antibodies, and those start becoming visible at about 7 days. And when we get antibody responses, this also is accompanied by inflammation, and we think that the inflammation is partly driven by the virus, but then what happens is that we think that the inflammation is then taken over by the host, us, potentially looking something like autoimmune diseases, as we all know. These are, again, conditions where the antibody response or the host inflammatory response goes out of control. And this may be driven by virus replication, we're still not sure. But that's your immune phase of the disease, and that happens from 10 days onwards. And then you either recover from this or things don't go well, and some patients do die, unfortunately. And this is accompanied by inflammation in the lungs, which leads to a picture that looks a little bit like ARDS, or acute respiratory distress syndrome. I know that, there are multiple organs involved. We won't really go into that. There's cardiac involvement, there's prothrombotic tendencies that may be related to immunity. We're not sure at the moment, but that's a general picture of how the time course goes. And this is a really unique. There are very few diseases that look like this. That's why this has been so fascinating, and why it's been so difficult to treat, because we've never really seen this kind of picture triggered by a virus in so many people. So just a bit more about spike protein here. We won't spend too long, but this is essentially what I was talking about earlier. This is the virus binding to the ACE2 receptor in your lungs or your nose. Once it binds, a number of other complicated things have to happen. Because the spike protein is made up of S1 and S2 fragments, this has to be cleaved by a protease, and this thing called TMPRSS2 is one of our proteases sitting around on our cell surface, and this cleaves this thing, this spike here. Once that cleavage or breakdown has occurred, you can then the virus is then able to bring its membrane close to our membrane, and this fusion peptide becomes active, and then causes a fusion of the membrane. So you can see that the surface membrane here fuses with this, and then therefore you've got direct access of the virus, whatever was inside the virus, into the cell. And this is a picture of just the RNA coming into a cell. It doesn't obviously come just like that, but so that's how the fusion and entry happens. And just to show you the importance of that entry mechanism for the survival and transmission of this virus. Now, you may not have heard of this mutation that arose in the spike protein, but it happened back in February this year, and this is just showing you the percentage of all the people with the mutation. So this is what happened before. So Wuhan strain came, back in December, and this is all Wuhan strain in orange. And then what happened is that this mutation occurred in China, but then rapidly spread around the world, such that now 99% of our infections are with the mutant form. And this mutation has been shown in experiments, and we've done some of these experiments to show that the virus is more able to replicate. It's faster, and it allows the virus to transmit better. So this is the importance of studying what's happening to the protein on the surface, because it can completely change the look of how this infection behaves. And of course, if the virus can do this in terms of just improving its own transmission, you have to think about what it would do when you start trying to use vaccines to stop infections. You can imagine the virus will simply switch to using different proteins or different amino acids, and then can escape antibodies. So this is the big worry right now. This is just another picture of that spike molecule, and these are two mutations that we found in a patient. I'll talk about them later on. But it just shows you that we can use structures. These structures are derived from techniques such as cryo-electron microscopy or even crystallography. These are disciplines in physical sciences and chemistry that have been around for quite a few years, and are absolutely essential to our understanding. And again, to highlight the basic sciences, the only reason we will survive this pandemic, and we must invest in science because it is really the only way out. So, immune responses. So just a little overview of what we know about immunity. This is going to be a very superficial look at it, so that we can then discuss COVID-19. So there are three types of immunity. One is called innate immunity, and this is basically the defense proteins in your cells, which have evolved with us since we were, even back before monkeys, way back into mammals, and even earlier. These are molecules that have developed over evolution. In fact, you can find some of these in fruit and flies, and we have these similar molecules that flies do, in fact. And what they do is they detect foreign substances. So they are there to detect pathogens such as viruses or any other sort of challenges. And what happens when the body detects these is they trigger interferon, essentially, or other inflammatory proteins, which make your body change in many different ways. It makes the cells much harder to infect, and it causes shutdown of certain processes in the cell. So this is something that we all have, and we think that some people who survive or who control infection may have slightly different innate immune responses, in that they can activate interferon very early and control virus. And there are some studies showing delayed interferon production in people who go on to have severe disease. But this is a bit contentious at the moment. Then you've got this adaptive immunity, and this is really what the kind of lay public know much more about, which is mainly because this is what vaccines are designed to generate. And this is adaptive because it's adapting to the specific pathogen. So the thing about innate immunity is it's a process that does not care about what the pathogen is. It is not specific to a virus or bacteria. It just is a general mechanism. Whereas adaptive immunity is something which your body is able to design specific antibodies or T cells which are programmed for a particular pathogen or strain of pathogen. So that's why we call it adaptive. So there are two types. One is mediated by B cells and that produce antibodies. And then you have these things called T cells, which essentially are lymphocytes which can kill infected target cells. And T cells are also responsible for us helping to control cancers before they become out of control. So vaccines, as I said, are designed to make both responses. And just a bit more about the T lymphocytes. You're eliminating intracellular microbes that can survive within your cells. But I think most of the talk will be about really humoral or B cell responses that generate antibodies. So you can see here, this is what we call a plasma cell. It's a type of B cell, a B lymphocyte. And once the B lymphocyte recognizes a particular pathogen, then it changes its shape and it starts producing large amounts of a particular antibody to control that virus. So the aim really is to produce these cells, and certainly nearly everybody who's infected with coronavirus will develop B cell responses and antibodies, but some will be better than others. This is a bit more detail about how T cells work, really. I mean, this may be slightly beyond scope, but essentially we have many patrolling cells in our bodies. These are kind of like as I say, the sort of secret service or police, as it were, cells like macrophages, which patrol your lungs and your nose, and they're looking for pathogens. And when the pathogen goes in, or is encountered, it's swallowed up by this cell, it's processed, and then specific proteins end up on the surface of that cell, or the antigen presenting cell. And what that does is engage specific T cells, which can recognize this. We generate billions of different types of T cells, each of which can respond to a specific protein on a specific virus. So we generate lots of different cells, and when they recognize the one that they want to kill, they will then proliferate, and you'll get huge numbers of expanded cells from this one cell that does the recognition. So this is the body's way of recognizing something and then hugely expanding its defense proteins. So T cells, again, can be split into two different types, CD4 and CD8 positive. We won't go too much into that, I think, because they have different functions. But moving on to antibodies, we all know that we measure serum antibodies. We used to call them sort of gamma globulin fractions, and some people have have deficiency in this, and I'll show you an example later on. But these are obviously very important, and we know there are 5 kinds: IgM, IgG, IgA, D, and E. They're made up of, this is what an antibody generally looks like, and this is the bit that will bind the protein of the virus. So this is the bit that will bind the spike of the coronavirus, for example. And this side of the antibody can then bind to cells. So that's a bit of a more complex function. This is how the B cell will then become activated via this section here, and other cells are also activated. So IgG, what's IgG? It's the thing that makes up most of your immunoglobulin. It's secreted in high quantities. It can go across the placenta, so this is what protects babies. So this is how maternal antibodies cross into babies. Its major functions are to neutralize microbes, sometimes toxins. There's this thing called opsonization, which is what I was mentioning earlier, where the IgG engages or binds to a cell, and the cell can then swallow up the protein or antigen that it has bound. So IgM, on the other hand, is something that's secreted earlier in infection. It's the first type of antibody that's produced, and it can't cross the placenta, and it is the early response. It's less specific, but it's there early. So in COVID-19, we've seen patients with IgM as early as 5 days after exposure, and then the IgG usually comes about 2 to 5 days after that. So the antibodies are very important, as I said, but then we have two types of use of antibodies, let's say, or immune therapies. We can either give them passively, which is where, for example, passive immunity is via breast milk in mothers, but also when we talk about use of convalescent plasma, which is what immune serum is, this is passive immunity, and you just inject antibodies into the person. The problem with that is that it obviously disappears, it's not maintained, and therefore it has a very limited sort of activity period. Active immunity, on the other hand, is where you can, is obviously what you get when you get a natural infection, but what we do with vaccination, this is what we're trying to do. We're trying to get active immunity artificially through vaccination, and I'll talk a bit about the ways we can do that. So types of vaccines. Let's talk, in the old days we had literally 1 or 2 types of vaccines. Now we have multiple types. Even I'm not an expert on vaccines, but I can talk a little bit about each of them. We have these things called live or live attenuated vaccines, which is where you take a real virus from a patient, let's say. You put the virus onto some cell lines, like cancer cell lines that we use in the lab to grow the virus. And as that virus grows in those artificial cells, it loses a lot of proteins which cause disease. So it switches off many of its sort of dangerous capabilities. And so that means that you get a less virulent form after it's called passaging. It means you're passing it through all these cell lines. So the advantage of making vaccines like this is that you're using the whole virus, and you often get the strongest immunity, because this virus, when injected into somebody, will cause a real infection. But of course, it will be less severe. So a lot of people like this, and certainly many vaccines have gone down this route. But the disadvantage, of course, is that you can get side effects. You can get reversion to wild type. The virus could potentially acquire its old properties. And one very good example of this, of a live attenuated vaccine, is of course the oral polio vaccine, which is a vaccine which replicates and is passed in the stool of children. And even if other people pick it up through contamination, they can get vaccinated as well. But once every 10,000 cases, you get real polio happening in the child, unfortunately. So that's the downside, that you can get actual disease. We don't think this is suitable for COVID-19 yet, because we haven't figured out, we don't have the animal models to show when the virus has become attenuated. So I think that's one of the main issues with attenuated vaccines for COVID-19. So other approach for a vaccine is a protein subunit. This is almost the simplest type of vaccine you can get. It's where you simply make the protein of a virus, like you can make the protein in bacteria, you can make the protein in other cells. You purify the protein, and then you put it into some suspension, and then you inject it into somebody. And a good example of this is hepatitis B, which uses the surface antigen protein of hep B. And you get a very nice immune response, nice antibodies, and it triggers B cells. So the disadvantage is that there's no real infection. You're just putting a protein in, so that it's just a protein reaction you're getting. So you only get partial immunity. Sometimes this is enough. For example, hep B, that generates enough antibodies to challenge the hepatitis B when the real thing comes into your body. And so this might actually work for coronavirus. The advantage is it's usually much safer, because there's no real infection, and you're only providing one protein, for example. We often need to use things called adjuvants, which you've probably heard of. These are things that are provided with protein, with vaccines, to stimulate the immune system, for example, to stimulate innate immunity. There's more evidence as time goes on that innate immunity is critical in helping the immune response, even through adaptive means. Then there are these things called viral vector DNA vaccines, and they're quite popular for SARS at the moment because they're kind of new, they're novel, and they have the advantage of encoding many proteins of the virus, even 5 or 6 proteins, and you put those proteins inside a common cold virus called adenovirus. And so the adenovirus infects the patient when you inject it into them, and then the adenovirus makes copies of itself, but it also makes copies of the SARS proteins, and then your body essentially is able to see these SARS proteins and generate an antibody response and a T-cell response based on this. So this has had very good results in vitro, although there are not very many successful vaccines. I mean, these have been used a lot for HIV and TB vaccines, but unfortunately none of these have really been proven to work. And I don't think there's any good example of a viral vector vaccine that actually works, but somebody may correct me. The disadvantage is that if you have had adenovirus in the past, you have your own antibodies, and when they inject this vaccine into you, you can pretty much, a number of people are able to just destroy that vaccine virus because they have antibodies. So what people have been doing is using a chimpanzee adenovirus. It's called ChIMPAD or ChAd, which you may have heard of, because our antibody responses will not kill the chimpanzee virus. But of course, there are numerous complications with using different platforms. Very briefly, RNA vaccines. This is a simple type of vaccine where you can just take the genetic code of a virus and literally the DNA or RNA building blocks of the virus, and you can encode them with inside vesicles or other mechanisms, and then you just directly inject this into the patient or the person, and this generates some good immune responses. So, for example, and the advantage is you can encode the whole virus, so you can put RNA encoding the entire virus in, and you'll get responses against each segment. That RNA will go into the cells and start making protein, and that's the aim. So you'll get a protein response as well. The disadvantages, of course, you may get side effects because RNA is a very powerful stimulus for the innate immune response. So it can stimulate quite a severe inflammatory reaction. And of course, our cells are designed to detect foreign RNA, so there are barriers to overcome that. So that's a sort of quick overview of vaccine technologies. These are 2 slides I'm just showing you on the pipeline. I mean, there's a huge number here. I'm not going to go through each one, but you can see if you look at the platform, for example, the one that people have been talking about is obviously Oxford's vaccine with AstraZeneca. This is using the viral vector, the chimpanzee adenovector that I was talking about, and that is expressing the spike protein. So that's one that people are very hopeful for, but there's a lot of hype about this. I'm not sure it's going to work. Then there are these RNA vaccines here. You can see more viral vector vaccines from Johnson & Johnson. Protein subunit vaccine here. Again, this is using SARS-CoV-2 glycoprotein, which is the spike. This is also spike. You can see this is why the spike was so important, because all of these vaccines are actually targeting this one part of the virus, which to me is a little bit worrying because it happens to also be the part that the virus can change very, very easily. This second page, there's a DNA vaccine here, which will make copies of virus proteins, and the list goes on and on. So you can see there's a huge number of vaccines in development. And sorry, just before I stop there, there are these whole inactivated vaccines here. So this is not attenuated, this is actually inactivated, so the virus will not replicate, it will just go in and activate the immune system. And that's one area I didn't really cover. And it's a sort of very traditional vaccine design. So whole inactivated, for example, pertussis vaccine is an inactivated vaccine. So a long list. Many are in phase 3. Let's hope some of them have some impact. The big problem with the vaccines is that, or the testing is that because we can't do human studies, we have to show protection in in areas where there's a lot of transmission. So that takes a lot of patience, it takes a lot of money and a lot of time. If we could do a challenge study and give somebody a vaccine and then give them the virus, we could test protection very easily. And some of those things are being planned, of course, but they are dangerous because we don't have any good treatments for COVID-19 at the moment. So let's talk just quickly about how you measure the efficacy of a vaccine. As I was mentioning, you need to show protection at population level. You need huge numbers. A challenge model would be great, but there are ethical issues. Animal challenge models are the other way that people have usually done these types of things, but the problem is there aren't any really good animal models for COVID-19. We are not really using monkey models very much at the moment. I'm not sure what the issues are there, but of course there are a lot of ethical issues, and these are expensive experiments. But one way we can do, and this is what a lot of people have been doing, is to measure the human response after a vaccine dose in the patient by taking blood. And what you do is you take the blood and you take the serum and you measure neutralization capability of the antibodies in the person after vaccination. And we've been doing some of these experiments in our lab, mainly to detect antibodies in people. And the nice thing, the way we use this is, in fact, this is also based around HIV. And this is what most people globally are using. So you take an HIV particle and you put essentially the spike protein on top of that particle. And so what you're measuring is the spike protein combined with the serum and antibodies in the patient. And if there are antibodies in the patient because of the vaccine, the antibodies will bind to the spike protein on the HIV particle, and it will stop that virus infecting these target cells in a plate here. So if you have no serum or no antibody, you're going to get a lot of infection by that little virus particle. If you put in lots of antibody or lots of serum, you can see in this patient here that the virus, the amount of virus infection goes down really dramatically. This is a log scale. So this is 100 million, 10 million, a million, and 100,000 infection events. So you get a massive drop just by putting in serum from a patient. And these are real data from real patients in our hospital, from a study we did recently. And you can see that this person here in blue has very little antibody response. So this person is negative for coronavirus antibodies, and this person is strongly positive for antibodies. And this is what you're trying to elicit with a vaccine. So these are live cases of infected patients, but when you test a vaccine, you need to be seeing this kind of response. And many of the vaccine studies have shown promising results in that they can show this happening in people. The question is, how long do these antibody responses last? And that's what's ongoing. These are the kind of curves you get, which I've seen. You can get 100% infection, and then it goes down as you put in more serum. And we've been doing a lot of experiments recently in our lab based on this, because we are interested in, my group is interested in virus mutation. We've worked on HIV drug resistance for many years, and essentially it's the same process that HIV uses to escape the drugs that we use. The reason we don't have a vaccine for HIV is because it's the most mutagenic virus in man. And making vaccines, therefore, is very difficult because the virus is always changing. But what about SARS-CoV-2? Although it has a mutation rate 10 times less than HIV, it still mutates quite a lot. It's an RNA virus, so there is mutation there, despite what people have been saying. And a very good example was the news in the last few days about the minks, the mutations found in minks, which have been infected by humans. And now these minks have infected humans again. And those mutations are being passed on to people now, which has worried the world quite a lot, because they've shown that these mutations make some of our monoclonal antibodies ineffective. So I'm going to show you just in the last few minutes just some stuff that we've been doing with a particular patient and how we use this to study virus evolution. So although people say mutations are slow, I'm going to show you a case where mutations are happening quite rapidly. So this is a patient who had immune suppression. There was a lymphoma diagnosis. The patient had chemotherapy, including a drug called rituximab, and this drug it kills your B cells. So because, of course, many diseases are driven by B cell dysfunction, and some cancers need B cells or are driven by B cell proliferation, and they need to be destroyed. So if you give somebody rituximab, you take away their B cells, you take away their antibodies. So this person had very little antibodies, or very few antibodies, and very poor T cell response. He unfortunately he went into hospital and acquired coronavirus in another hospital in Cambridgeshire, sadly, by infection from someone else. And then he came to Cambridge. He went into ITU with shortness of breath and needed some oxygen, given some remdesivir. And then, because things were not getting better and there was very little antibody, we had to give him convalescent plasma here in July. Then things got worse again. And then he had more remdesivir, more plasma. But unfortunately, he progressed and multi-organ failure, pneumonitis, and then death, basically, in August. So obviously that's a bit of a sad story, but we had a study going on at the time, and we collected about 23 different samples from his nose and throat and tracheal aspirates, where we have been able to sequence the virus because we have quite sophisticated sequencing here. And we've been able to track what was happening to the virus. And here, this is what we call a phylogenetic tree. You can see, where do we start? Okay, so on the right here, you can see all the sequence. These are sequences from around the UK. You can see that they're fairly uniform. And whenever you get these branches, this is where the virus is mutating. So anywhere you see a long branch is mutation. You can see this patient has quite long branches here compared to everybody else that's really kind of close together. This patient has started expanding. And if you blow this up in a way or show us in a different way, this is one patient here, and these are 5 other patients. 1, 2, 3. Sorry, this is 1, 2, 3 control patients. You can see their virus sequences from different time points are very close to each other. So these are all different sequences and time points. But this guy has, look at this, it's a very long, very, very wide distribution of sequences, showing that the virus has been evolving. This is another way of showing the same information. This is the green patient here. There are these major lineages arising. The virus is really changing a lot in this person. And we think this was driven by the plasma that was injected into him, and it was possibly driven by remdesivir. Because if you can't clear the virus is making a lot of copies of itself, it's mutating, and therefore it's changing a lot. And you can see this in these phylogenetic trees. We've made some viruses that have some of these mutations in them to test how they behave in the lab. For example, this mutation here, P330S, makes the virus much less effective at infecting, so it causes a defect in the virus. But we think this probably must be accompanied by some advantage for the virus, because otherwise the virus would not acquire this mutation for no reason. So this mutation must be doing something in the patient. So we're currently trying to figure out what that is. We've done some antibody escape assays, but these are sort of ongoing at the moment. But I think that's probably where we should stop. And there are a lot of people who contributed to the work we've been doing and some of the information I've showed you. And we can now open for discussion.
Thank you for your talk. Before opening question and answer to the audience, I like to make a couple of remarks. Before you joined, Shalini Winston told me you are the top 10 urologists across the world. Is that right?
Virologist.
Virologist, yeah. Okay, virologist. And I didn't want to tell you in the beginning because that may skew your talk.
I don't know if I'm one of the top 10, but
now on behalf of MSMA committee and members, I agree you are the top 10 virologist in the world.
And he is another top 3 Indians recognized in the world by Times.
Okay, excellent. There we are. There is a second one comes from Amar. Then your earlier slides, I'm a surgeon. I'm scared of reading even ECG. That's why I chose surgery. I didn't become a physician. Yeah. So your earlier slides only remind me of noodles mixed with another noodles. I didn't understand anything. but the question I put forward is, okay, let me go through the basics. Last week, I looked at Guardian newspaper, and it said there's a 13 agents on phase 3 trials, but none of them is marketed to the public community yet. But then since yesterday, I hear news that vaccine will be available to GPs across UK from 13th of November, that's next week, and it's given in 2 doses. So what's they talking about? What I am missing here?
Yeah, so I mean, there's a bit of a rush to get these vaccines available, and this would never happen in a normal circumstance because there is no large population data for any of the vaccines yet. So what they're saying is that because of the experiments that they've done in the lab, they've given volunteers these vaccines, and they've measured the amounts of antibodies in response to the vaccine. They are saying, therefore, we can predict that this will protect you. Now, so there are a lot of things that assumptions they've made. But the biggest assumption is that there will be lasting effects of these antibodies in you. And we've seen from some quite big studies that even if you're naturally infected, your antibody levels do decline over time. So the question will be whether these vaccines, how often you have to give them, how long will you be protected for? Well, these are questions we don't know the answer to. But because of desperation, I think that, and also I think the need to show that something's being done, because they did so much hyping. The scientists in Oxford, for example, who I disagree with a lot, and a lot of these people made too many promises too early. I mean, they were talking about 6 months to make a vaccine, which misled a lot of people. So I think part of the response now is because they have to show that something is happening.
So the analogy is you're treating the pre-diabetes. So actual treatment of diabetes is not available yet. Is that right?
Possibly, yeah. Yes. But it will give a chance to study the responses. I mean, in a way, this is a natural experiment. So I think